So whether or not an action potential is generated depends upon the balance of excitation and inhibition into the system. So the balance of excitatory postsynaptic potentials caused by excitatory neurotransmitters and inhibitory postsynaptic potentials caused by inhibitory neurotransmitters. So what we're going to see is that each neuron has a number of inputs, both excitatory and inhibitory. The neuron will decide on the balance of excitation and inhibition, whether it's going to fire or not, and that's called synaptic integration. We'll probably cover this a little bit more in the subsequent screencast. So let's just maybe in this screencast consider excitatory synapses in a little bit more detail. A very good example is acetylcholine. And acetylcholine acts at what we call cholinergic synapses. So acetylcholine is packaged into vesicles and is released in response to an action potential causing an influx of calcium through these voltage-gated calcium channels, as I showed you maybe in the previous screencast. Acetylcholine vesicles fuse with the membrane. Acetylcholine is released across this narrow synaptic cleft and interacts with different receptors. So as I said before, some of these receptors are excitatory, for example, the nicotinic receptor. Muscarinic receptors can be inhibitory or excitatory, depending on the subtype. We'll see as we go through later on in the course, maybe there are 5 different types of muscarinic receptors. Some of them cause an excitation, some of them cause an inhibition. And that's briefly just due to the fact that they couple to different pathways downstream of acetylcholine binding to the muscarinic acetylcholine receptor. So these are the ways in which acetylcholine can signal. It can open what we call an ionotropic receptor, a nicotinic receptor. It lets sodium down its concentration gradient and causes a depolarization. We'll consider this in a little bit more detail over the next few slides. Elsewhere in the course, we'll consider what we call G protein coupled receptors, and we'll see that they signal differently to these ion channel coupled receptors. So let's consider the acetylcholine receptor in a little bit more detail, just to show you how we open a nicotinic acetylcholine receptor, how we let sodium down the concentration gradient. So a little bit like the voltage-gated channels that we talked about when we considered the nerve action potential, our nicotinic acetylcholine receptor at rest has a gate, and the gate is closed. So there's no movement of ions, in this case sodium, across the membrane. So the receptor is said to be inactive or at rest. What happens when acetylcholine comes in? Acetylcholine fits into its binding site, usually on the outside, an extracellular region of the receptor, and causes the gate to open, essentially. So acetylcholine going in to its binding site opens the gate. Sodium, which is at higher concentration outside the cell, comes down its concentration gradient and causes a depolarization of the cell and excites the cell because the inside becomes more and more positive. So sodium entry depolarizes the postsynaptic membrane and causes an excitatory postsynaptic potential. So it's very important here not to get confused between the nicotinic acetylcholine receptor or receptors in general and voltage-gated sodium channels. So remember, the important difference is that the channels that we talked about when we were talking about the action potential are different to these receptors. They have got a gate, but in the action potential, the gate is opened by voltage. Here, the gate is opened by a chemical, sometimes called a ligand. So you might hear these called ligand-gated channels, and that lets sodium into the cell. So remember that this is different to the action potential. So it causes what we call an excitatory postsynaptic potential. And again, we'll describe these so that you know the difference between an EPSP and an AP, an action potential. generates an action potential if the threshold is reached. So an EPSP is something that leads to the generation of an action potential. So if we consider this, what we can do, there's a branch of science called electrophysiology, which we do in the lab that I work in. And electrophysiology essentially measures electrical activity, in this case of the presynaptic neuron, shown here, and the postsynaptic neuron. So what we can first see here is the action potential is always looks the same, effectively. Yeah. So this action potential is the same in height and duration as this one and this one, whereas the EPSP can be very different. So first of all, it's smaller than the action potential, but you can see it varies in size. So this one is smaller than this one is slightly bigger, and these two can add together. Action potentials can't add together, they can't get any bigger, they can't get any wider, but EPSPs can. So there are differences between these action potentials in the presynaptic neuron and the postsynaptic EPSPs. So let's just consider some of the properties of the EPSPs to show you why they're different to action potentials. So first of all, well, compared to all or nothing action potentials, EPSPs are smaller. So you can see that for a start. These go from -70 up to above 0. So, you know, around about 80, 90 millivolts. Here, much smaller, around about 10 millivolts, -70 to -60, slightly smaller, slightly bigger because these two have added together. So these three properties that I've described here can be said to be graded, temporal, and an EPSP is able to summate. So graded means that they can vary in amplitude. So as I said, this one is slightly bigger than this one. These two added together are bigger than this one. They're temporal. That means they decay with distance. So you can see that this decays over time. Maybe this decay is similar, but can be slower, and this decays over time. Whereas the action potential decays very quickly, around about a millisecond. Here you can see the scale down here. So the EPSPs are longer events essentially, and they can add together or they can summate, which the action potential cannot do. So again, take home message, remember the difference between EPSPs and action potentials. The waveform looks very different. Action potentials are formed in the pre-synapse. EPSP, as their name suggests, forms in the post-synapse. Okay. So if we talk about summation, this idea that I gave you back on maybe the first slide, what we're looking at with summation is the amount of excitatory versus inhibitory inputs. So on this slide, we'll just consider excitation. When we look at inhibitory neurotransmitters, we'll consider inhibitory and we'll consider summation in a little bit more detail. So as I said, this is our excitatory terminal coming in. These are vesicles of some excitatory transmitter. We could say acetylcholine, it could be glutamate. Essentially, it's probably useful to call it acetylcholine here because we're saying that when we open the receptor on the postsynaptic membrane, we get sodium influx, and we know that's true for acetylcholine. As the inside of the cell gets more and more positive, so remember that we said that the neuron usually sits around about between -70 and -90. As we get more and more positive charge, it gets more and more positive. So it moves away from the resting potential, goes more and more depolarized. So say to here on this, we've labeled it round about -45. What happens when this gets depolarized enough, we reach a threshold and the action potential fires. So that's said here, stated here, transmitter release at a sufficient number of presynaptic terminals simultaneous is needed to occur simultaneously in order to pass the threshold that maybe we saw on one of the previous slides about the action potential, and an action potential can fire. And again, remember from this diagram that I showed you previously, the action potential gets generated at this area here, the axon hillock, sometimes called the axon initial segment, as it is on this diagram. This is just a diagram from a different textbook. And the action potential can spread down the axon to these to the next terminal. And so information can be transmitted from this pre-synapse to this post-synapse on down to the next pre-synapse in a chain, effectively. So yeah, this is just again a placeholder slide, just to remind you that this is the threshold that has to be overcome in order for the action potential to fire. So again, if we go back here, the action potential is generated here once a threshold is reached. So this resting neuron, in this case around about -65, is depolarized by about 20 millivolts. That is sufficient to overcome the threshold here. You can see the threshold is you know, if you were to measure that, it was probably be around about maybe around about minus let's say -45, because that will fit with the previous diagram. If the threshold is reached, the action potential can fire. Okay. So again, maybe that is that's a nice place to finish this screencast. What we'll do in the next screencast is talk about inhibitory synapses.